Thank you everyone for being here today. My name is Hussein Al-Haribi. I am master student at Department of Electrical and Computer Engineering, King Abdulaziz University, Saudi Arabia. I'm very happy to present my research work in this first National Student Conference on Spectroscopy, and I am honored to share my study result with you about assessment of conjunctive task by functional near-infrared spectroscopy. In this presentation, outlines are introduction, research background, methodology, result, discussion, and conclusion. In this introduction, I am going to talk about the functional near-infrared spectroscopy (FNIR) is an optical-based technique useful in the field of neuroscience studies. There is a growing interest for this tool in neuroimaging, brain activation, psychological investigation, and behavioral science. FNIR measures hemodynamic response induced from a task. Near-infrared light can pass deeper into the biological tissue and human head as well. Based on the light absorption by the tissue, the change in concentration of oxyhemoglobin and deoxyhemoglobin can be estimated using modified Beer-Lambert law. The FNIR has found its application in conjunctive and behavior studies. The FNIR has several advantages over other neuroimaging modalities. FNIR has better spatial resolution and less affected by noise rather than EEG. It is portable, cheaper. In the methodology, measurements were recorded from the FNIRS system. The data was analyzed from NIRS SBM software tool. For the specific conjunctive task, the block model and the event relay model were used. As the target area of the conjunctive task experiment were within the primary prefrontal area. This task is within the limit of the penetration depth of NIRS. During the task period, subject was asked to perform given task. In this study, we selected 10 healthy volunteers with their consent. FNIRS data was collected from a Lebanon system Shimazu Company, Japan, at King Abdulaziz University, Jeddah, KSA. We used 6 transmitter fibers and 6 receiver fibers with total 15 channels. The processing of the measured data was done with NIRS SBM software. The conjunctive task given to the subject was watching a video clip. Here, as you see, the Lebanon system Shimazu was used. The left picture display recordings from selected channels, results, and discussion. As we can see here, the channel for number 14 showing the raw data collecting from subject. On The blue one is processing data after we applied GLM for the signal. As we can see here, during the time task, we notice that oxyhemoglobin is higher than deoxy. This graph showing region of interest analysis by average channels. As we can see in the beginning, the deoxyhemoglobin is higher than oxy, and this means the subjects are in the rest. After that, starting the oxyhemoglobin increasing. To conclude this work, we found that the oxyhemoglobin was higher than the deoxyhemoglobin concentration. Thank you.